Now next we will talk about the diagnosis of genetic disorder. How you will diagnose a genetic disorder. We mainly diagnose the genetic disorder with the help of the pedigree chart. For understanding the pedigree chart, you need to understand certain symbols. Okay. So the first one is a circle which represents a female. Next one is a square which represents your male. This one basically represents your unknown gender. Okay, this means that the gender of that baby is not known. So this was all about the genders. Now see, in this you are able to appreciate that there is some black things inside the circles also and inside the square also. This means that the individuals are diseased. This means the individuals are diseased. Okay. Next thing that you are able to appreciate is a thin line between the two individuals. What does that represent? That represents that there was a marriage. This represents that there was a marriage between the two. And see, there are two lines. What does two lines means? Two lines means that there was a consanguineous marriage. There was a consanguineous marriage. What does the consanguineous marriage means? Consanguineous marriage is basically a marriage who are between the close relatives. Okay. And here the autosomal recessive disorders are high. Okay. Next thing that you have is you are able to see that there is a female and there is a male and they both had marriage, they both had their offsprings and they have the dizygotic twins. Next, male, female, both had their marriage, but you are able to see that there is no offspring. This means that this is basically a case of infertility. Infertility or no kids or no offsprings. Next that you are able to appreciate the half circle is affected and half square is affected and half of them are not affected. So this means that they are basically the carrier of the disease. And in the second figure you are able to appreciate that there is a arrow. What does arrow represents? Arrow basically represent that this is a index case. An index case, you will learn more about index cases in your community medicine lectures. Okay. Now see this figure. In this figure you are able to appreciate that there are two arrows which are crossing through the circles and through the square. This represent that this is a death due to disease. This represent that it is a death due to disease. And in this one there is a circle and inside the circle there is one more circle which represents that there is X-linked carrier. Which represent that there is X-linked carrier. Now next you are able to appreciate a female, a male. They had their marriage. Oh wait, in the marriage section you are able to appreciate that there is a break. What does this mean? This means that they are divorced. Okay, this means the individuals are divorced. Okay, female, male had their marriage, had their intercourse, had their healthy offsprings. This represents the offsprings. Okay, next thing that you have, female, male had their marriage. Oh wait, this one is similar to the dizygotic twins one, right? No, it's not similar. See, the difference is here. Here you are able to see the two different things that is happening. So it is the monozygotic twin and that was a dizygotic twin. Now next thing that you need to understand is about the inheritance pattern. Inheritance pattern. What are the inheritance pattern that we have? We have the autosomal dominant, autosomal recessive, X-linked dominant and X-linked recessive, right? So first let's learn about the autosomal dominant. Now normally what does the word dominant means? The word dominant means that there is a dominant person. Like there is a dominant person in your home. Who is a dominant person in your home? Is mainly your mother. She is a dominant person. So what she do? She gives everyone orders that you have to keep the house clean, right? And everyone obeys her because they want the food, right? So similarly, in the autosomal dominant, you are able to see that the males are affected, okay? The females are also affected. So we are able to appreciate that there is no gender bias, okay? Your mom gives food equally to your brother and to your sisters, okay? So in autosomal dominant, there is no gender bias, and you are able to appreciate that the first generation is also affected and the second generation is also affected. So this means that there is no skipping of generation. There is no skipping of generation that is there, which means the all generations are affected. Okay. Next that you have is about the autosomal recessive. Autosomal recessive. Like suppose you have a elder brother, okay, and you are the younger brother. So who will dominate more? Your elder brother will dominate, and you are not able to raise your voice because he is elder to you. Similarly, in the autosomal recessive one, you are able to appreciate. What you will be able to appreciate, I will show you in a second, okay? You will be able to appreciate, see, this is the offspring they had, okay? Then they had their marriage, they had their healthy offsprings. Now they had their marriage. What is happening? They had a affected offspring. They have the affected offspring. You are able to appreciate one thing. In the first generation, is there anyone who is affected? No. In the second generation, is there anyone who is affected? No. But in the third generation, there is effect. So you can conclude that there is no gender bias. Why? Because the females are also affected and the males are also affected. So there is no gender bias. And but one thing is there is skipping of generation. There is skipping of generation that is present. So this represents your autosomal recessive disorder. Next that you have is about the X-linked recessive. For the X-linked recessive, draw the pedigree with me. You have a female, you have a male, the male is affected. Then they had their offsprings. What does this represent? This represents that it is a carrier stage. It represents that it is the carrier. Okay. Next thing is female is a carrier, had a marriage, other offsprings are also there. Female is a carrier, male is not affected. Then they had their marriage. What is the outcome of that marriage? Female becomes a carrier, normal female and a affected males. So you are able to appreciate one thing, that only the males are affected, right? And it's the harsh truth of this universe as well, that males are more affected. compared to the females. Although I'm a female, I should not say this. But still, yeah. So the males are more commonly affected. Males are affected more. And the females are basically the carrier. And the female are basically the carrier. Now next thing that you have is about the X-linked dominant. Next that we will talk about is the X-linked dominant. Now what happens in the X-linked dominant? In X-linked dominant, there is a female, there is a male. Male is affected, okay? Because mostly males are only affected, not females, okay? Again, in the second generation, the male is affected. They had their children and they had their marriage. See, what will happen in the second generation is that the males will not be affected. The males will not be affected. It is the daughters that will be affected. It is the daughters that will be affected. The males will remain healthy. Okay. So you are able to appreciate a pattern that in the X-linked dominant, the male was affected. Now, what should ideally happen? The disease should go 50-50, hand in hand. That is 50% to the female, 50% to the males. But what is happening? All the trauma is given to their daughters, because that is also the harsh reality of this society, that all the traumas are given to the daughters only. Okay, so you can see that it is the disease from the dads to daughter. Okay, and there is no skipping of generation. Although it's not that important to know that whether there is a skipping of generation is there or not, but this thing you need to keep in mind, that if the disease is starting from the male then and proceeding further to the females, which makes them affected, so this means this is a case of a X-linked dominant disorder. Next thing that you have is about the mitochondrial inheritance. And I have told you earlier also the mnemonic of M and M. M and M means mitochondria means mother to all. It is a disease which is transmitted from mother to all the offsprings. Okay, I will also draw a pedigree for your comfort. Female affected, then the offspring, female affected, male affected, female normal, then the offsprings, you have the female affected and a male affected. So you are able to appreciate the pattern that female was affected, female transmitted the disease to all the female transmitted disease to all the offsprings. Okay, to all the offsprings. So this was about the inheritance pattern. Let's just recap. We have seen the symbols that are used for the pedigree analysis. You have male, female, unknown gender, disease, marriage, consanguineous marriage, dizygotic twins, infertility, monozygotic twins, offsprings, divorce, X-linked carrier, death due to disease, index case and carriers. And we have talked about the inheritance patterns, that is autosomal dominant. In autosomal dominant, there will be no skipping of generation that will be there. In autosomal recessive, there will be skipping of generation that will be there. In X-linked recessive, the males are affected and females are basically the carriers. In the X-linked dominant, the males are affected and there is no skipping of generation. The disease is transmitted from dads to all the daughters. And in the mitochondrial inheritance, you have M and M pattern. That means mitochondria means the disease is transmitted from mother to all the offsprings. We are ready to go. to the next topic, that is your karyotyping. What does karyotyping means? Karyotyping means a study of chromosome. Karyotyping means the study of chromosomes. Now, I have told you when we were talking about the Down syndrome that you can diagnose a patient of a newborn of Down syndromes antenatally as well. Okay, antenatally means through the pregnancy. So, we have the two scenarios when we are doing the first step, that is the sample collection. What are the two steps we have? It can be either a pregnant female or it can be a non-pregnant female. For the pregnant woman, what you do? For the pregnant woman, you basically have two types. Either you can do the chronic villus sampling or you can do the amniocentesis. Chronic villus sampling is basically done at the 10th week. Amniocentesis is done in between 16th to 18th week. Okay, and they are done antenatally, and more you will be able to study when you are studying your obstetrics and gynecology. Now, if the female is non-pregnant or a grown-up female, then what you will do? You will do the blood sampling. And with blood sampling, you can do the skin biopsy. Okay, either you can do the blood sampling or either you can do the skin biopsy. Now, second thing that you need to understand is the chromosome arrest. So, chromosome basically arrest at the metaphase stage with the help of 𝗲𝗵. What is 𝗲𝗵? 𝗲𝗵 is basically a plant derivative. Okay, and the chromosomes are basically best visualized at this stage. The chromosomes are best visualized at this stage. Third thing that you have is about the fixative. For the fixative, you have a special fixative, that is the Karyotyping, okay, that you use. Fourth step is the trypsin digestion. And they basically help to remove the unwanted protein, and they basically help to remove the unwanted proteins. Okay, till now, am I clear? You have sample collection. For the pregnant woman, you do the chronic villus sampling and amniocentesis. For the non-pregnant or the grown-ups, you do the blood sampling or skin biopsy. In chromosomes are mainly arrested in the metaphase stage, and here the chromosomes are best visualized. For the fixative, you have the Karyotyping, and the trypsin digestion basically helps to remove the unwanted protein. And the lastly that you have is the banding. Okay, what does banding means? Banding means that if the chromosomes are A and T rich, okay, that is basically a sequence. If they are A and T rich, they will have a dark banding. And if they are C and G rich, 𝗧𝗲𝗹𝗵 rich, then they will have the light band. Now, if we are talking about the bands, let's just talk about the types of banding that we have. Okay, so I'm writing, let me just write it over here again. Banding. First one is the G banding. G means the 𝗲𝗼𝗄 stain. 𝗲𝗼𝗄 stain with light microscope. 𝗲𝗼𝗄 stain with light microscope. Then you have the R stain. This means reverse banding. Reverse banding with light microscope. Then you have the T banding. This means the telomere part of chromosomes with light microscope. And then you have the C banding. This means the centromere part. Q banding. Q is basically for your a special stain called as the quinacridine stain with the fluorescent microscope. Okay, so this was all about the banding technique that we have. We have G banding, R banding, T banding, C banding and Q banding mainly. Now next that we will be talking about is the arrangement of chromosome. And guys, you have to draw it with me, okay, because if you will draw with me now, you will be able to remember more. If you will draw with me, you will be able to remember. And guys, tell me how many pairs of chromosomes we have. How many total pair of chromosomes we have? Do we have 20, 21 or what? Like this, you have to tell me. It's actually very basic and I know most of them don't know, but still I am asking you. You have to tell me and draw this diagram with me, okay? There is no excuse. If you will draw now, you will able to remember each and every thing. And this will help you in future as well. This is chromosome number 13. This is chromosome number 14. Chromosome number 13 is used for if there is a deletion in the chromosome number 13, then this means it is a case of Patau syndrome because Patau the peoples at the age of 13 years. Okay. Next one is your 19. Then you have the 20, 21, 22 and anything else left? Yes. You have the X and Y as well. Okay. Now we are good to go. Look at this. This is the proper arrangement of your chromosomes. Now I am giving you certain examples. If I told you that there is a deletion at 5 P chromosomes. There is a deletion at 5P chromosomes. What does that represent? That represents that it is a case of Karyotyping syndrome. This represents that it is a case of Karyotyping syndrome. Now, next if I told you there is a deletion of 13 number chromosome or there is a trisomy of 13, then what does it represent? It represents your Patau syndrome. Okay, so you have to see each and every chromosome very well. If there is a trisomy of 18, then it is a case of Edward syndrome because we have seen the mnemonic E for E. What is the age at which you are able to participate in the process of election? That is the age 18. And E for E, election is E and Edward is also E. Next, very important. Trisomy 21. What does it mean? It means a case of Down syndrome. Okay, now tell me guys, in Down syndromes, what you have in the palms? I have told you what we have in the palms. We have a Simpson's crease. What does that mean? There is a single crease, not like many creases that you and I have in our hands. Okay, so this was all about the chromosomal pattern. If there is a deletion of Y chromosome over here, if I write there is XO, then this becomes the case of your Turner syndrome. And guys, in Turner syndrome, I have given you the classic clinical features. What are they? They are the webbing of neck that will be present, and mainly the presenting one will be a female, and the counterpart of male is the Noonan syndrome. Okay, next that I want you all to understand is about a technique called as a fish technique. What does fish technique means? Fish technique is nothing but a chromosomal abnormality. But it is mainly a chromosomal abnormality technique. Okay. See, this is the figure, and in this you have certain red lights and you have certain green lights. Green lights are less as compared to the red one. So you are able to appreciate that this will be your chromosome number 13, which is presented with the green color, and this is the chromosome 21, which is represented with the red color, and the red colors are more. So the red colors of chromosome 21 are more, and we all know that 21 means the Down's syndrome. 21 means the Down syndrome. So see, fish is not that much important from your university exam point of view, but it can be asked in your MCQs that are asked in your university exams. Okay, so one more thing, I can give you an example of the breast cancer. In breast cancer, you have the HER2/neu amplification. You have the HER2/neu amplification. Okay. Next thing that we will be studying is about the PCR. What does PCR means? PCR means polymerase chain reaction. Okay. And it is the amplification of DNA. The steps that are involved is, first thing that you have is the denaturation of DNA, means split the double stranded, split in the double stranded at 94 degrees Celsius. Second step that you have is the annealing of primer. This happens at 54 degrees Celsius. Next that you have is the extension. For the extension, there is converting the single strand DNA to double stranded DNA. Okay. Now, mainly there are three steps involved. We have seen there was denaturation, there was annealing, and there was extension. Okay, have I made this much clear? What are the three steps that we have? Denaturation, annealing, and extension. Now let's talk about the types. We have the types of PCR. What are the types of PCR that we have? First one is the Sanger sequence, and Sanger sequence is the gold standard. Sanger sequence is the gold standard. Next that you have is the SBPE. What does SBPE means? It means single base primer extension. Okay, single base primer extension. And the example is the hairy cell leukemia. And the example is the hairy cell leukemia. Third that you have is the RFLP. What does RFLP means? RFLP means restriction, fragment, length, polymorphism. And it is basically for the unknown locus. Next that you have is the reverse transcription PCR, and that is basically for RNA virus, like for the COVID, and here the DNA is amplified, and here the DNA is amplified. Fifth one that you have is the qPCR. What does qPCR means? qPCR means the quantitative PCR or the real-time PCR. It is for the COVID, for the HIV, and for the Epstein-Barr virus. Okay, next thing that you have is the RT-PCR that we mainly do for the COVID, for the COVID-19. Next on plate that we have is the GWAS. What does GWAS means? It means the genome wide association study. It is mainly for your research purpose. And the examples is diabetes mellitus and hypertension. Okay. Next technique that we have is the pyrosequencing. Next that you have is the pyrosequencing. Pyrosequencing is basically for the contaminated sampling. It is basically used for sampling the contaminated things. Okay, it is basically a better technique. Next that you have is the nested PCR. What does nested PCR means? In nested PCR, we mainly use two primers. The two primers are used and it is more specific. Okay. Next that you have is the droplet PCR. For droplet PCR, what you have? You have the DNA, then the small droplet, and then the PCR. Okay, so this was all about the types of PCR that we have. Let me just write it over here also, the sequence that we follow. We first have the denaturation, then we have the annealing using multiple probes. Right? Then you have the ligation, and at last you have the amplification. So this is the sequence that we follow and you need to keep in mind. Okay. Next thing that you have is about the CGH. What does CGH mean? CGH means comparative genomic hybridization. Comparative genomic hybridization. What does this mean? This means that there is a hybridization of parent's gene with normal gene. Okay, there is a hybridization of parent's gene with a normal gene. Okay, whether it is diseased or not, it's not a matter. For example, you have certain color coding for this. Red one are basically the test gene. Green one are your control gene. Like if I draw right now with you all the square, then if I draw, you have certain test genes, then you have certain control genes. Okay, and normally let's take these are the normal ones. Okay, so what is happening here? You have the red genes, right? Then there is a gain of function. Then there are blue genes, normal, and the green one, there is a loss of function. Okay, so this was the basic mechanism that it follows. Okay, I hope I made it clear. Let's just learn about the PCR. PCR can be asked as your short note. Okay, it can be asked as your short note question. So PCR is basically the amplification of DNA. It has three processes: denaturation, annealing, followed by the extension. What are the types of PCR that we have? We have first one the Sanger sequence, that is the gold standard. Then we have the SBPE, that is the single base primer extension. Then we have the restriction fragment length polymorphism, RT-PCR, qPCR, GWAS, pyrosequencing, nested PCR, and droplet PCR. So these are the types of PCR that we have. And in the last also we have given that there is first denaturation, then there is annealing which uses the multiple probes, then there is ligation, and then the amplification. Lastly, we have talked about the comparative genomic hybridization. Comparative genomic hybridization, we have talked about it. This means that there is a hybridization of parents' gene, whether they are normal or not, with the normal gene. And in this figure, we have represented blue with the normal one, red when there is a gain of function, and green when there is a loss of function. So, this was all about the PCR and the comparative genomic hybridization. Okay, so we have learned about the pedigree analysis, right? We have learned that there is autosomal dominant or autosomal recessive, X-linked dominant, X-linked recessive. Okay, so I think now you are able to understand and you will be able to solve the questions that will be asked on that topic. So I am giving you certain questions. We will solve that together. Okay. The questions are basically to identify the inheritance pattern. Identify inheritance pattern. You have to identify the inheritance pattern. So, are we ready to go? Okay. And you all have to give me the answers of this, right? So, the first question that you have a female, then you have a male which was affected. Then they had their offsprings in which male is okay, female is affected, then the female is affected, male is okay, female is affected. What is the first thing you will do? First thing is that you will find out what are the affected things that we are getting. Male is affected. Male is affected. See the first generation. In first generation, are you able to appreciate that the males are normal? And only the females are affected. Daughters are given all the traumas. So this is a case of X-linked dominant. This is a case of X-linked dominant. Next question that I am giving you is, here the female will be affected. Male is normal, they both had their marriage. Then they decided to have their offsprings. Here the male is affected, okay, it's cool. They had only, let me just draw it, only one offspring. The one offspring, the male is affected. They had their marriage with a female which was not affected. They had their offsprings in which mainly the female is affected. Mainly the female is affected. What is your diagnosis? I am giving you options also so that you are not able to confuse, because I know many of them are thinking that it is mother that is affected and mother is transmitting the diseases to all the generation. Okay, so I am not giving you that it is a case of mitochondrial inheritance. I am giving you the options as the autosomal dominant, autosomal recessive, X-linked dominant, or X-linked recessive. So you will not start with this thing, first one. You will start looking from here. From here you are able to see the male is affected and the male is transmitting all the diseases to the females. Male is transmitting all the diseases to the females, means the male is giving trauma. Male is giving trauma to his daughter. Male is giving trauma to his daughter. This means this is a case of X-linked dominant. This means that it is a case of X-linked dominant. Okay, now the third question. Third question that you have a female and you have a male. They both had their marriage, but the male was affected. Then they had their offsprings. In the offsprings, the male was affected again. Decided to have their marriage. The female was not affected. Then had their offsprings. What is this a case of? Again, you can say that it is a autosomal dominant. Autosomal dominant. Is it a autosomal dominant case? No, I'll tell you why. See, everyone is given 25% weightage out of 100. 25, 25, 25, 25. In this generation, out of 100%, only 25 is affected. So how it can be a case of autosomal dominant? It cannot be a case of autosomal dominant, because in autosomal dominant condition there is a 50-50%. 50% males are affected and 50% females are affected. So it's not a case of autosomal dominant. Is it a case of autosomal recessive? No. Why it's not a case of autosomal recessive? Why it is not a case of autosomal recessive? Because we have seen that in autosomal recessive, there is skipping of generation that is there. There is skipping of generation that is there. And in this figure, there is no skip of generation. So it is not autosomal recessive also. Now we will look into this. Here, the males are giving trauma to all the females. So again, it is a case of X-linked dominant. Okay, so again, it is a case of X-linked dominant. Now I'll give you a last pedigree. Okay, this will be your question number 4 and a last pedigree. You have a female, then you have a male. The male was affected. Then they had their offsprings. Okay, then they had their offsprings, out of which almost everyone is affected. Had their marriage, affected, affected, not affected. Will this be a case of X-linked dominant? No. I'll tell you why it is a case of autosomal dominant. I will tell you why it is a case of autosomal dominant. See, here the males are affected. Okay. Second thing that we are able to appreciate is there is no skipping of generation, that is there is no skipping of generation. Next that we are able to appreciate is out of 4 offspring, 25%, 25%, 25%, 25%, out of 100%, 75% out of 100 is affected. This means it is a case of autosomal dominant, and there is no skipping of generation is there and no gender bias is there. So that's why I'm very confident that this is a case of autosomal dominant. If there would have been a skipping of generation, then it can be a case of autosomal recessive. Okay, so this is how you have to do the pedigree analysis. Each and every step you have to take into your consideration. If you will do the pedigree analysis by this method, it will be very easier for you all guys to do the pedigree analysis. Okay, so this was all about the pedigree analysis.